eventually. Let's look at these checkpoint questions quickly. So I'm going to go over these. Again, these are topics, some of these at least, are things we talked about in lab. So for example, the order of the meninges from outermost is going to be the dura mater, the arachnoid mater, and then the pia mater. The second question, what is CSF? Where is it found? Where is it synthesized? Where is it absorbed? So CSF, or cerebrospinal fluid, is the watery clear fluid. It bathes our central nervous tissue. It also helps to regulate a unique environment around our central nervous tissue that's different from the plasma, what's happening in the rest of the body. It is synthesized on the ependymal cells that line the choroid plexus, which is really the central part of the lateral ventricles. It circulates through the lateral ventricles, third ventricle, fourth ventricle, and then all the way around the spinal cord and brain. And then it's finally reabsorbed back into the venous system via the arachnoid villi, right? The villi that we spoke about. Okay, what is the function of the blood-brain barrier? So really, simply, the blood-brain barrier protects the nervous tissue and neurons from the exposure of the environment in our blood. Any toxins in our blood, pathogens in our blood, harmful metabolites or drugs that are in our blood cannot cross the blood-brain barrier and cannot affect the nervous tissue. So it's a way to prevent or protect the delicate brain tissue from harmful exposure. What anatomical structure between the endothelial cells limit the diffusion of water-soluble molecules? So here we talked about the tight junctions of the endothelial cells, as well as the foot processes of the astrocytes that provide this, this distinct separation or this protection of the nervous tissue from what's happening inside the capillaries, right? It prevents certain substances from leaving the capillaries, especially large substances like proteins, and again, harmful substances, bacteria, etc. Okay, so someone's asking me to post the checkpoint questions, and I can do that. So I'll post them in Brightspace after our session. All right. Any other questions from this topic? So we've got about 10 or so minutes left, and I'm going to kind of shift gears here and talk about some of the topics that okay, so someone else is asking if this material is going to be on the exam. Yes, it will. So everything up until this point will be on the exam. Okay, so I'm going to switch gears here and I'm going to go over and talk about some of the topics that I got for clarification. So one of them had to do, hang on here. Okay, so one topic that I got was kind of clarifying the equilibrium potential of sodium and potassium and how we generate this resting membrane potential. So I'm going to jump back to this drawing that we did. That kind of was a summary of the establishment of the resting membrane potential and just talk about how the equilibrium potential of our ions relates to this resting membrane potential. So the equilibrium potential, let's kind of start with the definition of equilibrium potential. This is the membrane potential at which our ions, respectively, are no longer moving. The chemical driving force and the electrical driving force are equal and opposite, and the ion is no longer moving. Now, it's going to be different for different ions. For potassium, it's -94. For sodium, it's +60. And so that's the membrane potential value at which these ions are at equilibrium. And so when we think about the chemical driving force, it's trying to get the particles to equilibrium. This is why the chemical driving force is reflective of the equilibrium potential. This is what that force is trying to do. It's trying to get the particle to that state where it's no longer moving and it's at equilibrium. The Membrane potential is really about the state of the cell, the charge on the particle, and the polarity of the cell. So the fact that we have this opposite attracting negative charge trying to move to the positive environment, positive charge trying to move to the negative environment, and because we have this polarity of the cell, we've got this separation of charge. This is another force that's also acting on our particle. So we've got two forces acting on our particles. One is the electrical force, one is the chemical force. And so we can look at these two forces separately, but when we think about the movement of the ion overall, we really want to bring both of those two forces together. And so for potassium, for example, it has a chemical driving force out of the cell. So in order for this to be at equilibrium, the direction of movement would be out of the cell according to that concentration gradient, but its electrical force is going to be into the cell, okay? Where it's a positive charge ion trying to move to the negatively charged environment on the inside of the cell. And so we see two different directions of the force. So we subtract those different values and we get a smaller force that's in the overall direction out of the cell. How do we know that the overall direction is out of the cell? Because we then compare the magnitude of the equilibrium potential to the magnitude of the membrane potential. The equilibrium potential is greater, and so the direction of the chemical driving force is the overall direction of the particle, which is out of the cell. Okay. For sodium, things are a lot more straightforward. Sodium has an inward electrical driving force, again, positively charged ion moving into the negative environment inside the cell. It also has an inward direction as far as its chemical driving force, because it's at a higher concentration on the outside of the cell and a lower concentration on the inside of the cell. And as we say that, we want to be thinking about the sodium-potassium pump in the background that's creating these two gradients, right? So the direction of sodium movement will absolutely be inward because both forces are inward. When we look at the magnitude of sodium, it's going to be a much stronger force because we're going to add these two forces because they're in the same direction. And so the overall direction will be strongly inward inside the cell. Now, when we relate this to the actual value of the equilibrium potentials of these ions, the ions are going to be moving or trying to move until they get to equilibrium. So this is the force that's acting on these particles. So they're always going to want to be moving in the direction that would bring them to equilibrium. They will never actually get to equilibrium. Why? Because the cell at rest is really favoring both of these ions. And so it doesn't settle on either equilibrium potential, but it kind of meets in the middle at -70. And it's not actually in the middle. Why? Because the equilibrium potential for potassium is closer because there are more potassium channels on the cell. So the resting membrane potential is going to look more like potassium's equilibrium potential. It's going to favor potassium equilibrium potential more because there are more channels on the membrane. There's more movement of potassium. All right. Now, if we completely removed all of the sodium channels, then potassium would eventually get to its equilibrium potential and vice versa. But because we have both the movement of both ions, then we never actually get to the equilibrium potential of either ion. We kind of move around in the middle and we settle at -70, which is closer to potassium because of that higher potassium permeability. All right, so kind of on this note, I've got a question here from SI. So this had to do with the osmolarity. So the question is, if you add 0.2 millimoles of sodium chloride and 0.2 millimoles of glucose to 1 liter of water, what is the osmolarity of the solution? The answer is 0.6 milliosmoles. Okay. So let's talk about why that is. So remember, sodium and chloride are going to dissociate, okay? So although we look at sodium and chloride collectively, when they're in solution, they're going to further dissociate into sodium and chloride respectively. So essentially we've got 0.2 millimoles of sodium, 0.2 millimoles of chloride, and then 0.2 millimoles of glucose combined in that 1 liter of solution. And that was how we would get that 0.6 milliosmol as far as the overall osmolarity. I hope I'm reading that question correctly. But I think that that's the answer. And you can follow up with me by email if if you want further clarification there. Okay, but essentially when we think about sodium chloride, it's going to further dissociate. Remember we said that the osmolarity is really a reflection of all of the solute particles into consideration. And so even though sodium chloride is one collective compound in solution, it's going to dissociate into sodium and chloride respectively. All right. Okay. Any other questions on this? Hopefully that kind of clarified the equilibrium potential and how we get that resting membrane potential, which is really a sharing of the equilibrium potential of both of those ions. All right. And then the last thing that I'm going to touch on here has to do with the synapse and the fast versus slow response. Again, I'm going to share a review video that kind of touches on this. So if you need more clarity, definitely look out for that video. But just to kind of go over the events at the synapse here, we've got the action potential traveling down the axon. Remember, it's because of the concentration of those voltage gated channels at the nodes of Ranvier. So it's kind of leaping down the length of the axon. It gets to the axon terminal. It triggers the opening up of the voltage gated calcium channels where calcium can rush into the axon terminal. It's going to then trigger the exocytosis of this vesicle. Remember, this is why we have that brief delay of this transmission, the transmission of this signal. And so these vesicles need to dock at the membrane here, fuse with the membrane of the axon terminal, and then dump or release that neurotransmitter into the cleft. That neurotransmitter will cross the junction, bind to its receptor, and this is where we have that fast versus slow response. In order to cease the signal, we then have to clear away the junction. So that's done in conjunction with these two molecules. So we've got the enzyme here that's degrading any excess neurotransmitter, as well as the reuptake molecule that's also providing a way to recycle that neurotransmitter and bring it back into the axon terminal and create a new synaptic vesicle. So we can also have this neurotransmitter diffuse away from the junction, or it can be degraded and diffused away as well. So a lot of options here as far as how we can clear the junction. The important thing is that we must clear the excess neurotransmitter in order to cease that signal. And then looking at the different types of responses here, again, comparing the fast to slow response, this is what happens on the membrane when that neurotransmitter binds to the receptor. So if it's an ionotropic receptor, this is a fast response because the receptor is also a channel. Now it's going to open up when that neurotransmitter binds, and depending on what type of channel it is, what ion it's allowing, that will determine if it's an EPSP or if it's an IPSP. And then we can also have a slow version of this response on that cell, and that's if the neurotransmitter binds to a metabotropic receptor. Now, metabotropic receptor is coupled to a G protein. That's kind of what slows the whole thing down, is that it's not an immediate response the way it is for the ionotropic receptor, because there are several players involved in the process here. So the G protein is really what kind of slows things down. We have to then dissociate a part of this protein off. It's going to activate an enzyme in the membrane, and then we're going to produce the second messenger that will help to transmit the signal to opening up the channel. So again, the signal can be transmitted by the production of the second messenger, either directly producing some type of enzyme that brings about that response, or the second messenger itself can then open up the channel that's downstream in the membrane. And again, depending on what type of channel we open or close, that will determine whether an IPSP or an EPSP is generated on that adjacent neuron, okay? And that's kind of the crux of the difference between the fast response and the slow response.